Hi, everybody. Welcome to Chapter 3. Today, we're going to be talking about the cell. So we're going to start off with general cell structure and the different types of cells that your body has inside of it. So we'll start off with this slide here, and this table shows us that there is cellular diversity all over your body. So we have cells that are going to connect different parts of our body or cover organs or also line them. We have cells that are going to move organs or body parts. We have cells that are going to store nutrients. And then we also have cells that are going to help fight disease, like add to your body's immune system. So this is some of the examples, right, in this column of cells that connect or cover or line organs. So these are called fibroblasts, right? These are going to help connect different parts of the body together. As you guys can see, they kind of branch out and they grab different things and they kind of pull them in. These here are going to be called simple cuboidal epithelial cells. They're going to line a surface or cover or line an organ. And then the last ones down here on the bottom, these are erythrocytes. The more common way to say that is red blood cells. And red blood cells, we've talked briefly about before, but they are going to help carry oxygen to the body and CO2 out. So O2 and CO2 out. In the second column, the cells that move organs and body parts, these are all muscle. So there's 3 different types of muscle. There's skeletal muscle, smooth muscle, and cardiac muscle. Now, the visible differences you guys see between them, you're going to learn about in lab. The functional differences we can talk about as well in lecture. So skeletal muscle, you can think about that type of muscle as anywhere you have voluntary or conscious control, right? So if I'm thinking like, I need to tie my shoes, right? That means I need to use muscles in my forearms and my fingers to go through the rhythm of tying my shoes. That's all skeletal because I'm consciously thinking about what I need to do and then moving those muscles. A smooth muscle is going to be found lining what we would call a visceral organ or something that is involuntary. So like you ate dinner and then you need to digest it, but to digest the food, you need to move it from the mouth, down the esophagus, into the stomach, into the small intestine, then the large intestine. That type of movement is going to be done with smooth muscle. That's a very good thing. Those muscles contract without you having any knowledge of it or putting any thought into it. So you don't have to think, oh, okay, now I swallowed my food. Now I need to move it physically down my esophagus. And then I need to move it into the stomach. And now it's time to move it into the small intestine, right? It happens naturally or involuntarily, which means it's lined with smooth muscle. You don't have to think about it. And then the last type down here is called cardiac. Cardiac muscle is also involuntary, but because it's called cardiac and cardia means heart, this is a involuntary muscle that is only found in the heart, which means your heart itself is a muscle. I personally think that's a really good thing because you have the ability to condition it, meaning make it more effective, because just like any other muscle, you can strengthen it, which means you can strengthen your heart and make it more efficient. The third column here, it says we have cells that store nutrients. So things like our fat cells, these help store fat for our bodies, because really your body doesn't know that you're consuming anything like food. You consume it for any other reason other than the fact that it is going to carry nutrients that you can convert into energy. So your body's under the assumption if you're consuming it, eventually you're going to convert it into energy. So it's trying to not get rid of too much of it. So you store it in fat cells. We also have cells that gather information and control body function. These are going to be called our nervous cells or nerve tissue, right? They're going to send and receive signals. So this would be a neuron. Cells that fight disease are going to be like macrophages, lymphocytes, monocytes, basophil cells. These are going to fight diseases and make sure that anything that's bad is being attacked and killed, and then anything that's good is going to be able to stay alive. And then the last part here is we have cells that are for reproduction. So males produce sperm and females produce eggs or oocytes, and those are to help reproduce. There's a lot of diversity in our body, as you guys can see, and each type of cell has a different function. So there's approximately 100 trillion cells in the body and 200 different types of cells. The general cell itself is composed of many different organelles inside of it. There's no single cell that includes all of the features that you see here. They just put all of the features in here so that you guys can identify what they would look like if it was a part of a cell. But again, like for example, the flagellum A flagella is like a tail. This is only found in sperm. So if you're a female, you do not have flagella in your body at all. And we can go through one by one and talk about, where each one of them would be found and things like that. But just know that the majority of cells do not have all of these features inside of them. So we'll go through and talk about the organelles in just a second. Cells are going to be divided or compartmented into three main parts for the ease of study. The first part is the nucleus. The second part is the cytoplasm. And the third part is the plasma or cell membrane. The nucleus is going to be the circular organelle in the center of the cell that's going to hold the DNA. The cytoplasm is going to be made up of the cytosol, which is the fluid in the cell, and then the organelles. And then the plasma membrane is going to be the like the protective layer that's going to divide the inside stuff from the outside stuff. So basically keeping all those organelles on the inside and then keeping everything else out. So again, the nucleus, most bodies, cells have a single nucleus. Some cells have none at all. So like a red blood cell actually gets rid of its nucleus. But then there's others that have several nuclei. We would call that multinucleated. So an example there would be a skeletal muscle fiber. There are different parts of the nucleus. You have a nuclear envelope, which is the outside portion of it. In the nuclear envelope, you have pores, which are little openings. And then the inside, you have DNA. This is your cell's genetic information that gets arranged into chromosomes. And then you have the nucleoli, which is the center of the cell. Okay, so one more time with the nucleus, it is going to contain DNA. Humans, so you and I, have 46 chromosomes. These genes are going to be found in linear, meaning straight arrangements on the chromosomes. And then the genes that are on the DNA are going to be the blueprint for a specific protein. So the protein is really, really, really important because the protein is going to help direct all of your cellular activity. So the blueprint for the protein comes from the gene, which is on the DNA, which tells you in a roundabout way the DNA in your body is going to eventually get to the point where it would code for a protein. So it's going to make you, you. Nucleus is going to be a non-dividing cell that contains chromatin. Chromatin is just another way to describe the genetic information because it's structured differently, but it's still DNA. So if I had to draw it, right, this would be chromatin, which is DNA, but we also have the ability to wrap it a little bit nicer and neater for when you go through the division process. You probably have seen something like that. This is the same thing as that in regards to its makeup. It's just wound differently. This looks like a jumbled up ball of yarn. This is a very nice, neat two-arm structure, which we call a chromosome. Chromatin, chromosomes, same genetic information, right? Same DNA here as you have here. It's just arranged differently. So again, in this case, this is called chromatin. And then this has the ability to divide equally. You just divide it down the center, and that's called chromosomes. So again, it's just condensed DNA, and you have two sister chromatids. Each one of these individually is called a chromatid, TID. Be careful when you guys are looking at these words because chromatin and chromatid are one letter difference, right? This is chromatin. One of these arms is a chromatid. And then the chromosomes are going to be held together at the center, which we would call the centromere or the waist of the chromosome. Chromosomes, again, long molecules of DNA that are coiled together around what we call histone proteins. So it looks like this in a double helical strand. Then you're going to start to wrap around this protein. And when you wrap a bunch of them around a protein, they start to coil up. And then eventually they're going to condense tighter and tighter and tighter until they reach this two-arm structure here. So the nucleus, you have DNA and chromatin. You have RNA, which is going to be in the nucleoli. You have a nucleoplasm, which is going to be the like cytoplasm of the nucleus. A nuclear envelope, which is around the outside. And then again, the pores, which are going to be embedded in the nuclear envelope. A ribosome is going to be a two subunit structure that is going to be in the nucleoplasm, specifically the nucleoli. And then in the cytoplasm or endoplasmic reticulum, which is basically just saying we have ribosomes all over the place because their function is to synthesize or create proteins. So the cytoplasm has different compartments, right? You have the cytosol, which is the liquid portion of the cell. So it'd be like that clear stuff right there. Then you have an endomembrane system, which is stuff on the inside. You have lysosomes, proteasomes, mitochondria, peroxisomes, and then parts of the cytoskeleton, which help retain the structure of the cell. So one by one, the cytosol is going to be the intracellular fluid. Because it's fluid, the majority of it is water. It has a lot of what we call inclusions and dissolved solutes, which basically means proteins, carbohydrates, lipids, and then inorganic substances, or if you guys recall back to the biochem portion, stuff without carbons. The chemicals in the cytosol are going to be either in solution or in colloid, which means they're going to be suspended. And then functionality, it is going to be the medium in which many metabolic reactions occur. So when we say, where does this thing occur? Oh, it occurs in the cytosol of the cell, right? That's the essentially intracellular fluid where lots of reactions occur. The endomembrane system, so you have the nuclear envelope. So again, this is the nucleus. This is the nuclear envelope, which is around the outside, and then it has the nuclear pores inside of it. And you have the endoplasmic reticulum, which would be here and here. So you have two different types of ER. You've got rough ER, which basically has bumps on it. You guys can see the bumps are those dots. And then you have smooth ER that has no bumps. Then you have the Golgi apparatus, and you have things like transport vesicles that are going to, they're going to like move towards the plasma membrane, and then they're going to fuse and let out or let in different substances. And then you also have lysosomes here, which are going to it's called proteolytic enzymes, which basically just helps, it means they help break down things. Okay, so clinical application here. One of the functions of smooth ER is to detoxify drugs. So repeated exposure to certain drugs produce changes to the smooth ER in the liver, which results in a tolerance to the drug. It's how addiction takes place, because you need more and more and more of the drug to elicit a similar response. So drug or alcohol. So alcohol stimulates the formation of what we call cytochrome P450-2E1, which basically just means this very hard to remember name is going to generate what we call free radicals, which we talked about again in biochem, through the oxidation of a nicotinamide or a tinamide, adenine dinucleotide phosphate. So that's a really fancy word for NADPH. So what happens is chronic alcohol exposure means over a long period of time is also going to activate hepatic, so hepatic means liver, macrophages, which kill off things that are bad, that are then going to produce tumor necrosis factor alphas. So basically what we're saying is when you're exposed to alcohol for a long period of time, your liver cells that are trying to fight off the bad things are actually going to develop tumors, and they're going to start to break down. The stress, so the oxidative stress is going to promote hepatic necrosis and apoptosis. So it's going to start to like get infected and then spontaneously die, which is what apoptosis means. So the free radicals are going to initiate lipid peroxidation, which causes inflammation and fibrosis. So it's causing your liver to be fatty, to be inflamed, and then to develop like thyroids. So this is what a healthy liver looks like. And this is what a liver looks like for someone who's overconsumed alcohol for a long period of time, right? Chronic alcohol exposure. It's a very drastic difference. Proteasomes. So proteasomes are organelles that help destroy any unneeded, damaged, or faulty proteins. They contain an enzyme that we call a protease, which is going to help cut or cleave proteins into small peptide units. These are thought to be a factor in several diseases. So failure of the ubiquitin proteasome system is known to be linked to Parkinson's disease. Your next organelle is the mitochondria. So mitochondria is singular, or sorry, plural. Mitochondrium is singular. These are very, very commonly talked about in biology classes because they are what we would call the ATP powerhouse, right? Or the powerhouse organelle of the cell. It's because they generate a lot of ATP, which is your body's form of energy. That's where, so again, the mitochondria is where you have a lot of ATP production occur. DNA and ribosomes are going to self-replicate in this organelle, and they're going to be actually circular with 37 genes. So increased numbers with cells needed for ATP. So if you have a cell in your body that's going to be utilized a ton, you're going to increase the number of mitochondria in that area so you can produce more energy there. You're also going to have these organelles have a double membrane. So if you guys can tell, they have this outer membrane here, which is the red line. And then you see the yellow line, how it kind of like zigzags in. That's going to be the inner membrane. So the inner membrane is really important because it's arranged into these folds, right? Those divots in are going to be called cristae. The cristae are important because it increases the surface area, which means there's more of it, so that you can undergo more cellular respiration to produce more ATP. That's really important there. Peroxisomes, so similar in structure to lysosomes, but they're going to be a little bit smaller. These ones are going to contain enzymes that are going to use molecular oxygen to oxidize different types of organic substances. So they're going to help contribute to normal metabolic breakdown of things like proteins and fats, oxidize toxic substances like alcohol or formaldehyde, and then also catalyze and decomposes hydrogen peroxide. The cytoskeleton of the cell is going to be a network of protein filaments. It again is going to help support and shape the cell. And if you guys remember back to the beginning, cellular structure is going to contribute drastically to cellular function. So you really, really, really want the cell to maintain its regular shape, which is done by the cytoskeleton. So different portions to the cytoskeleton, you have microfilaments that are going to have actin filaments in them. They're helpful with movement and structural support. Then you have the intermediate filaments here that are going to be from this mid portion that are going to be for structural support, and then also cell-to-cell junctions, which means they help communicate to each other. And then the inner portion here are going to be these microtubules. They are produced by a tubulin protein that are going to help generate and retain cell shape, as well as transportation of the organelles. Okay, so your next organelles are cilia and flagella. These ones are really specific and only found in a couple of different places, but they both help with movement. So cilia are going to be numerous, short, hair-like projections, or sometimes people say finger-like projections that extend from the surface of the cell. They're going to increase surface area and help move material. Flagella are very, very unique, again, because they are only attached to a sperm cell. They're what you would call the tail, and they help the sperm move. Okay, so talking specifically now about the plasma membrane, this is a flexible barrier that is going to surround and contain the cytoplasm of the cell. This is what we would call a fluid mosaic model, which is this right here. And you guys can tell that it's going to be made up of what we call a phospholipid bilayer with different proteins embedded inside of it. So if you look at a cell and you pull out a portion of the cell membrane, you can see that there's a red layer here and a red layer here with yellow in the center. When you look more closely here, the red is here and here with the yellow in the center. The reason why you have that present is because you have a head and a tail. The head is the circular portion that is red. The tail is the yellow lengthy portion that is going to be in the middle. So the head here and here are both pointed towards the outside, and those are what we would call hydrophilic. Hydrophilic means water loving. So they're good to interact with the water. The tails are going to be what we would call hydrophobic. This is what you would call water fearing or hating, if you will. So they want nothing to do with water, and they only want to interact with each other. So they're pointed inward towards each other. Both of them put together make a phospholipid bilayer, which is the plasma membrane. And then you guys can see that embedded in that plasma membrane, you have proteins. These proteins are going to be channels that are used for transportation, or sometimes like the peripheral proteins, meaning on the outside, are going to be used as receptors and things like that. So again, framework, phospholipid bilayer. You have the phospholipid. You also have some cholesterol that's going to be embedded in the plasma membrane on the outside. It really just makes things a little bit stickier and easier to adhere to the phospholipid bilayer or the plasma membrane. And then again, you have protein channels through them. Okay, so every cell membrane has a mixed composition, again, of phospholipids, glycolipids, sterols, and proteins. Most of these proteins can move laterally through the lipid bilayer, so back and forth, and some of them form complexes that are actually going to move together. Lastly, some of them are going to be anchored to the cytoskeleton elements and stay in place, static. We have integral proteins that are going to be a part of this fluid mosaic model. Some of them, again, help with adhesion, so cell-to-cell connection. Some of them help with communication, which means they open channels to connect cells together. Some of them, again, are going to be receptors, like I said just a minute ago. And then others are going to help with cell recognition. So they're going to help tell other cells what they are so they can function properly to do whatever they need to. Okay, so these integral proteins that are going to be embedded in the phospholipid bilayer have the ability to aid in transportation. There's a couple different types of transportation. One of them is called passive transportation. The other one is called active transportation. So when you talk about passive transportation, you're talking about a channel that is going to be selective for a particular ion or molecule to move through, right? Like this is the only thing that can move through because I recognize it. Then you have others that are going to be carriers. They're selective for a particular ion or model. They're going to bind, kind of. And then you have proteins that are going to be undergoing active transportation. These protein pumps are going to require energy to move the material, either because there's a lot of it, or they have charges, or you're going against a concentration gradient. And then the last part is proteins are enzymes. They have the ability to be enzymes, which means they carry out metabolic reactions. I know we talked about enzymes in the biomolecule portion of it. So if you guys remember that they catalyze reactions. So what happens here is an enzyme would come and bind, and it would speed up the reaction of the reactants getting converted into the product. Okay, so transportation is really, really big in regards to the cell. And again, there's different types. So transport across the PM is plasma membrane. There it's going to be differentially permeable. Differentially just means it changes. Permeable means what can go through. So transport through the plasma membrane changes dependent upon how easy or hard it is to move through, which we'll start talking about here. So any small non-charged molecule like water or alcohol or O2, those can diffuse, meaning move through very, very, very easily. Anything that is going to be bigger, charged, or an ion cannot move through easily. So passive transportation, by definition, is not going to require energy. Active transportation, by definition, is going to require energy to move the stuff. So passive transportation, a couple examples are diffusion and facilitated diffusion, or facilitated transport. Active transportation, a couple examples here are like exocytosis or endocytosis. Passive transportation on its own gets divided into diffusion, osmosis, and facilitated diffusion. So here we're just going to talk about diffusion and facilitated transportation, which is also called facilitated diffusion. Both of them are going to require a concentration gradient. So basically what we mean with a concentration gradient, right? If you're thinking about a gradient, it means that you have like a variety of amounts. So with a concentration gradient, you're going to have more on one side than the other. So if we talk about, in this example, they have different types of solutes, meaning solids. But like, let's say that the purple is sugar, the yellow is potassium, and the The gray bubble is salt, right? So all of these things are solids, and there's more on the blue side than there is on the yellow side. When you talk about moving down a concentration gradient, the molecules will always move from an area of high concentration to an area of low concentration. So if we jump back here, we see that there's 1, 2, 3, 4, 5, 6, 7, 8, 9 of these solutes on the blue side and 3 on the yellow side. Because you're going to move from high to low, the molecules are going to move from the blue side into the yellow side. So they're going to diffuse through the phospholipid bilayer. But one thing that you can note is that the yellow molecules, we said were sugar, and they're bigger. Because they're bigger, they're going to need a little bit of help. They don't need energy because they can move from high to low concentration, but they do need some help because they can't go directly through the plasma membrane. So in this case, what happens is you use facilitated diffusion, right? Facilitating means like a helper, right? If you're going to facilitate a camp or you're going to facilitate an exam, right? You're going to go help there. So the facilitated diffusion means you have a helper. That helper is a protein channel. It's going to open up and make a bigger hole for that thing to move through so it can get from one side to the other. When you look at, I think we called this one potassium, I believe. The yellow was potassium, or you can call it water, whatever you want to call it. Basically, what we're saying is it's small enough and it's nonpolar with no charge, and it can move directly through the phospholipid bilayer. That is simple diffusion, right? It's going to move down a concentration gradient, but it doesn't need any extra help. So diffusion, again, small non-charged molecules like CO2, O2, water, glycerol, alcohol, they can diffuse very easily, and they will always do so until an equilibrium is met. So when you're looking at this image down below, you see that there's a lot of this molecule. We'll call it water. On the top side, you've got a phospholipid bilayer, and then on the bottom side, you have none. So over time, what's going to happen is the water is going to move from the top to the bottom. And then over a longer period of time, you're going to meet what we call equilibrium, meaning you have the same amount of molecule on the top as you do the bottom, or you could say same amount of water on the outside as you do the inside. That's an equilibrium. That's when diffusion stops. Facilitated transportation is very, very similar. It's still passive because you're just moving down the concentration gradient, but again, you just need those channels. It's going to do the same thing in regards to diffusion over time. It's going to try to hit an equilibrium. Okay, so that's diffusion. What is osmosis? Osmosis is a little bit different than diffusion. So it is actually when you have the diffusion of water across a differentially permeable membrane, or you would say a semi-permeable membrane. Semi-permeable membrane basically just means some stuff can get through and other stuff has to stay out. What is going to determine what stays in and what stays out and what moves through? Size. So if you think about it in this regard, osmosis has this semi-permeable membrane that essentially acts as like a coffee filter. Like a coffee filter is going to separate things based on size. Same idea with the plasma membrane. If it's small enough to get through, it will. If it's too big, it's going to have to stay on the other side. So osmosis is going to require us to learn 2 new terms. The first one is called a solute. S-O-L-U-T-E. This is going to be a dissolved substance, which would be like salt or sugar or calcium, right? It's a solid. And then the second term is a solvent, V-E-N-T. This is going to be the thing that does the dissolving. And in most cases, we'll talk about water doing the dissolving. So we could say the pink is water, and then the purple is, we'll just say sugar. So when we talk about osmosis, it's different also than diffusion because it's not everything gets to move or anything gets to move. It's only the solvent that does the moving. So you'll notice that you've got 1, 2, 3, 4, 5, 6, 7 sugar molecules in the beaker on the left side. you still end up with 1, 2, 3, 4, 5, 6, 7 after you're done. Hopefully, you'll also notice that the volume changed drastically, which tells you that the only thing that moved was the solvent or the liquid. So again, the diffusion or the movement of water across a differentially permeable membrane, and it's going to move until you also hit an equilibrium. So it's very important to identify here that equilibrium does not mean equal volume. It's most of the time going to mean unequal volume because the ions inside are going to differ in their concentration. So what's going to happen again is you're going to have a change in volume until the solute concentration is at equilibrium. So the reason that's so important is because when you have the movement of water in and out of the cell, it's going to change the structure. When you change the structure, you change the function. This is what we would call tonicity. So tonicity is going to be the concentration of solutes in a solution. And there's 3 different types of tonicity. In this example, we're using red blood cells to look at different tonicities. A red blood cell naturally is what we would call this biconcave disc. So it's not like a spherical cell. It has this like almost donut shape depression in the center. It's not a complete hole through, but it's a definite divot in the center. That's how it wants to look. So we'll start with the ideal environment. This is called an isotonic environment. This cell is happy. The reason it's happy is it's got an equal amount of water going into the cell as it does going out of the cell. That's a great thing because then you have the ability to keep the concentrations equal, which means that you're going to keep its shape and keep its function. Good, happy face. Then you have a hypertonic environment. I'm going to change my color here to see if we can make this a little bit easier to look at. So in a hypertonic environment, when you're looking at the solute versus the solvent, a hypertonic environment, hyper, hopefully you guys know, means a lot of. You have a lot of solute on the outside of the cell. So this would be like you ate in a giant bag of family-sized potato chips in one set, sitting or setting it. Basically what we're saying is there's lots of salt in your body on the outside of the cells, but very little on the inside of your red blood cells. What's going to happen over time is the solute or the solid is going to need to be, it's going to try to retain or remain in an equilibrium state. And because there's so much on the outside, the water from the inside of the cells is going to pour out. When the water pours out of the cells, that normal biconcave disc is going to shrivel in and it's going to undergo what we call crenation. Crenation is shriveling and it's bad because the cell eventually is going to shrivel too much and it's going to die. That's bad. And then the last example over here is opposite of this. This is going to be what we call a hypotonic environment. This is when you have a lot of solute or solids on the inside of the cell and very, very, very little on the outside of the cell. So this is a super drastic example, but it's a true example as well. What would happen if you drank gallons and gallons of deionized water, right? Purified water, very, very, very quickly. It is all bad. So can't remember what university it is, but it was probably 3 or 4 years ago where freshmen were being hazed, and instead of having them drink beer, they had them drink water really fast. They learned very quickly that that is not a good thing. It's not healthy and it's not okay. What happened is because the individual who was consuming the water drank too much deionized water too quickly, his outside environment of the cells, so it's called the ECF, had very little solute in it because he consumed so much water. So the concentration of that went way down. And because of that, the inside of the cells had a lot of solute in comparison to the outside, So what happened is the body tried to compensate. And instead of having too much on the inside, the body was pulling all the water from the outside environment to the inside of the cells to try to equilibrate that. And eventually what happened is the cells were getting bigger and bigger and bigger. And then they were going to undergo, we call lysis, like this one is doing, and it's literally going to burst. Cells that are bursting lose their structure, which means they lose their function, which means they're dead. Cells put together make tissues, tissues put together make organs, organs put together make organ systems, and organ systems put together make organisms. So eventually what's going to happen is the cells die, the tissues die, the organs die, the organ systems die, and then eventually the organism or the human will die. It can become fatal, which it did to him. That was very sad case, but he actually died from consuming too much water too fast. So that's a hypotonic environment. Now, don't stress out too much, right? That's a very extreme case. Your body has lots of feedback mechanisms that are going to kick into play. So if you eat a whole bag of potato chips or you drink a gallon of water, you're not going to die. Your body will adapt to it. Okay, that is passive transportation. Now we're going to move into active transportation. So active transportation is going to require energy. The molecules are going to move from a region of low concentration to high concentration. So the best illustration I can give you guys here is passive transportation is if you're thinking about a hill, right? And you're trying to go up this hill or down the hill. Passive transportation is going down the hill. Doesn't require a lot. You could literally just roll down. Active transportation is going to be going up a concentration gradient, and it's going to be very difficult to go up that hill. And because you're going against the concentration, we'll just say concentration here is 5, concentration here is 30, right? Normally you want to go down the concentration gradient. We'll just say it's like that. Passive is down, active is going against it. Because it's going against the concentration gradient, you have to use energy. So it's going to be opposite of diffusion, meaning that you're moving against the concentration gradient. So first type of active transportation is called primary active transport. This is going to use a protein carrier. This is the most prevalent type of active transportation. And it is seen a lot all over the nervous system in what we call sodium potassium pumps. It is going to require approximately 40% of all of your cellular ATP to remain operational. All of your cells have these, and they maintain low concentrations of sodium and high concentrations of potassium in the cytosol to keep these pumps moving. Next example is secondary active transportation. In this case, you have energy that is stored in the form of sodium or hydrogen ions in a concentration gradient that's going to be used to drive other substances against their own concentration gradient. So these are going to be things like antiports or symporters. So anti means opposite. It means something is moving in and another thing is moving out. Sim means same. So the two things are both moving the same direction. Either they're both moving in or they're both moving out. And then the last part is called a uniport. Uni means one. So you have one thing either moving in or moving out. And then the last type is called bulk transportation. Bulk transportation is literally what it sounds like. It means that you're moving a lot of things at one time. So it's like when you have a transport vesicle that's going to fuse with the plasma membrane, and it's going to open up and it's going to release or expel its substances into the ECF around it. Because you're moving a large amount of things, it's going to require energy. Endocytosis is exactly the opposite, right? The plasma membrane is going to, it's called invaginate or take substances in. And it's, again, a lot of substances. And so because of that, it's going to require ATP. Together, both endocytosis and exocytosis are going to help maintain total surface area of the plasma membrane because they have to fuse with it. So this is an example of endocytosis. You have something maybe like phagocytosis or pinocytosis or receptor-mediated endocytosis. All of it is taking something in, right? So you're going to kind of cleave around this particular thing and then engulf it. You're going to then complete the other side, and then you're going to create this vesicle or thing that helps transport, and you're going to move it into the cell. So this is phagocytosis, where you're going to essentially kill something. Penocytosis is like water drinking, and then receptor-mediated endocytosis means that you have to have receptors to bind to. Okay, next portion here is going to be cell surface specialization. So cells do not have the ability to exist alone, right? They have what we call a matrix, which is intracellular material in the cells that are going to be embedded. So this is also what you would call a ground substance. So here you have cell junctions that have molecular structures embedded into the plasma membrane that allow for things like communication or structural support. With communication or junctions, they have different types of junctions. So you have tight junctions, adhering junctions, or gap junctions. Tight junctions are going to be those that help seal connections that are usually found in epithelia. Then you have adhering junctions that are going to be used as structural components where movement is stretching the tissue. And then lastly, you have gap junctions that are going to be connections between the cytoplasm of adjacent cells that help allow for fast movement of material. So these are very specialized junctions. Here you guys can see a picture of a tight junction where you have these transmembrane proteins that are going to run from one side of the cell to the other. Gap junctions are going to be these pores that open through. And then anchoring junctions look like this, where they're going to be pulling from both sides. Anchoring junctions have a couple different types. They have what you call desmosomes, adherens, or hemidesmosomes, which basically just means they're taking place in different locations of the cell. You guys can see here, they're kind of at the midpoint of the cell. For desmosomes, adherens, they're a little bit more superficial. And then for hemidesmosomes, they're going to be found at the base of the cell. But all of them, again, are anchoring. Okay, so cellular communication. Cellular communication is really important. All basic communication happens like this. You have a target cell that's going to have a specific receptor for the signal. Then you have a combination of signals and receptor proteins that are going to cause a change at the target cell. And then you have a mechanism that has to be in place to turn off the action of that signal or the chemical. So signal transduction is how you convert a chemical signal into an intracellular action. So a chemical that's coming in into some sort of action that your body is going to do. So signal transduction is going to require a couple things. The first is a ligand. The ligand is going to be the first messenger that's going to then bind to the receptor. When the receptor is bound to, it's going to jumpstart the production of a response through a second messenger system or a process that we call transcription and translation. All of it, though, is going to produce a protein, which is going to elicit a response. And all of it's going to be regulated by responses for homeostasis. The way that we have the ability to communicate these signals is either local or long distances. Local means that they're going to happen close to the site. So through things like gap junctions, contact-dependent signaling, autocrine signals, or paracrine. So autocrine, auto means same. So autocrine signals are going to be of the same cell, right? And then paracrine is going to be, para means around. So these signals are going to go to cells that are right around the cell that released the signal. Then we also have the ability to communicate over long distances. These long distances are going to use different systems. They're going to utilize your endocrine system and or your nervous system to take these signals over long distances. Okay, so target cell receptors, you have the ability to have intracellular target cell receptors or in the cell membrane. So intracellularly, you have two types. You have lipophilic molecules that can diffuse through the membranes, and receptors in the cytosol or the nucleus. And then cell membranes, you have four types. You have the ability to have a receptor channel, a receptor enzyme, something specific called a G protein coupled receptor, or an integrin receptor. So basic transduction, again, you have a ligand that is going to bind to, so the ligand is yellow, that's going to bind to a receptor protein that is blue, and it's going to jumpstart a second messenger system, which is going to start what we call an enzyme cascade. You guys can think of this as like a domino effect. So the domino effect means once it gets started, they all push the other ones down, or when one enzyme gets started, it's going to start the reaction for every enzyme to occur. which is going to cause amplification of proteins and then release a protein to migrate towards a target and elicit a response. That's how transduction or cell-to-cellular communication occurs all the time with those basic signals. So this is getting a little bit more specific now, right? So now we have the ligand in yellow labeled, binding to the receptor, which is this pinkish color in the cell membrane. It's going to jumpstart what we call amplifier enzymes, which is the enzyme cascade, and phosphorylate proteins to produce an intracellular response, meaning it caused some sort of effect to occur. I'm aware that these concepts might be a little bit of a jump, so my suggestion is take it slow and back up and listen to it again if you need to, because we're going to move on to the next type now. So the next type is called a lipophilic hormone mechanism. This is where you have steroid and non-steroidal families that are going to essentially do the same thing, but in a little bit of a different way. So you're going to have a lipophilic signal molecule or a ligand that's going to diffuse through the cell membrane. When it goes through the cell membrane, now it has the ability to bind to a receptor in the cytosol, or it can go all the way into the nucleus and bind to a receptor in the nucleus. When that happens, it's going to jumpstart the process of transcription and then translation, which will create a protein. That protein, again, can then carry out the cellular response. It's really interesting to note that this is a slower response related to changes in gene activity. So while it has less steps than this example did, this one actually occurs faster because it has amplifier enzymes. So there's a ton of enzymes that are going to be working to create the phosphorylation of proteins. However, because enzymes speed up the reaction, the process here, while it has more steps, actually occurs faster. This one only has a ligand bind to a receptor, transcription, translation, proteins. That's it. But because it happens in a bunch of different locations and it's related to gene activity of the DNA, it takes a little bit longer. Okay, next part is the lipophilic ligand signaling amplification. So this just says everything that I just told you, right? Signal amplification, you're going to have one signal molecule result in multiple mRNA molecules. The mRNA then is going to result in protein formation, and that's going to then create a cellular response. So really what they just said is the process of converting DNA into RNA is transcription. I'm not going to write that out actually because that's going to take me forever. But this is transcription and then this is translation, right? So this is rewriting and then understanding how you wrote it differently, which is going to create the proteins. So this is an area where you can talk about physiological dosages versus pharmacological doses. Anytime you talk about the difference between what your body naturally creates, which is physiological, and what we produce in a pharmacy, which is going to be orders of magnitude higher to elicit the same response, it's always be much higher because when your body doesn't create it, it doesn't happen as naturally. So you're going to kind of force your body into responding to this thing that you're inputting in there. But because you're forcing your body to do that, you have to intake more of it. Okay, so then we have hydrophilic hormone mechanisms. So this is where you have ligands that are going to be water soluble. They require receptors in the plasma membrane. So you can't go through it now. You have to have them in the plasma membrane. So here you have signal transduction mechanisms and 4 different steps. So you've got the ligand or the hormone, which is green. It's going to bind to the receptor in the plasma membrane. And then it's going to transduce or activate the membrane receptor, which is going to activate what we call a second messenger system like cyclic AMP, or we call it CAMP. That's then going to jumpstart this enzyme cascade or the domino effect, which is going to produce a output or an effect that's going to hit a target cell and create a response, right? That's another way that you can communicate through cells. The actions of the hydrophilic ligands, this is simple. They open ion channels. They're going to help increase intracellular calcium concentration, which is going to change enzymatic activity. You can do that with enzymes called protein kinase or PK. or protein phosphatase. And again, it changes the calcium concentration or enzyme controls. You can do that through metabolic enzymes, muscle contractions, gene expression, protein synthesis, or membrane transportation / receptors, because it's how they bind to each other. So again, other ways that you're going to have these communication signaling going on. Next is a plasma membrane receptor. So the rapid cellular responding to enzymatic pathway, again, you have a ligand that's going to bind to a receptor, which is embedded in the plasma membrane, which is then going to jumpstart some sort of process that's going to produce a protein to create a response. There's 4 different categories of membrane receptors. One of them is going to be a channel receptor, which just means that there's a site for the receptor in the protein channel. Next one's going to be a receptor enzyme complex, which just means that one binds to the other. The next one's going to be a G protein coupled receptor, which means you have the ligand binding to a specific protein. It kind of looks like this like old like heating mechanism that you would see in like houses back east or something. And it's like a coiled thing. That's a specific protein because it's attached to the G protein on the intracellular side. And then the last one here is going to be an integrin. It's where you have the ligand bind to this tooth-like looking thing. That's going to change the cytoskeleton structure on the inside. So again, this is type 1. This is ligand gated. When we say ligand gated, anything that's a gate. So if you think about this like a door to your house, this is where you would have the door closed, right? When the ligand is closed, it means that nothing can move through. When you have the when the channel is closed, it means that nothing can move through. When you have the ligand bind to the receptor, what's going to happen is it's going to alter polarity and it's going to open up that channel, which means stuff can now move through. Next is the enzyme linked receptor. So an example here of the enzyme linked receptor is what we would call a tyrosine kinase receptor, or TK. Here you have transduction. That's going to be done through phosphorylation of the integral membrane proteins, which means on the inside, that lead to the regulation of other cellular processes. So it's going to control other things, right? It's going to activate enzyme phosphorylation and create proteins for a second messenger system, right? Phosphorylation of proteins. Then you carry out a cellular response. Next one is the G protein coupled receptor. So this is a 3-part transducer. It has a hormone or a ligand that's going to bind to the protein. To the receptor. Then you have the G protein on the inside portion that's actually going to pull apart, or what we would call break apart. It's called dissociation, right? It's going to break apart into an alpha subunit. And then that alpha subunit is going to bind to an enzyme that we call adenylate cyclase. ATP is going to be converted now into cyclic ANP and also 2 inorganic phosphates. They kind of flip those. So when that happens, it's energy essentially. And CAMP is going to be then activating the metabolic enzymatic pathways to open the ion channels or alter the enzyme activity to produce the proteins to carry out the response. Last one here is the integrin receptor. It's going to be using integral proteins in the plasma membrane to anchor the proteins intracellularly and change the cytoskeleton structure. Okay, last parts here is gene expression. Gene expression is really important because it helps a lot of the processes that we just talked about, plus it also codes for DNA and helps replication and all kinds of things. So gene expression is the information coded in DNA that acts as a blueprint for the production of polypeptides. Information transfer to create the gene expression is going to use this process. You're going to convert DNA into RNA, which again is called transcription. And then the last part here is RNA into proteins, which is now called translation. There we go. I knew I could type in here. So this is transcription. And see if we can, let's make this longer, get all the way over here. This portion here is going to be called translation. So transcription and translation. DNA into RNA is transcription. RNA into proteins is translation. So the whole process is going to start with replication. In order to divide and then rewrite and then understand in a different way, you have to first reproduce. So replication is going to produce a copy of DNA. This is going to happen before each cellular division. Then you have transcription. So transcription is going to be the formation of mRNA to take the genetic code from DNA, and this process occurs inside of the nucleus. Lastly, you have translation, which is going to convert RNA, specifically messenger RNA, to create them into proteins, and that process takes place in the cytoplasm of the cell. Cytoplasm. Oh man. Okay, in class, we can watch a video on gene expression. It's a very complex process, but I can give you guys a really quick overview of it so you guys can kind of see what that looks like in the process of replication, transcription, and translation. My link doesn't work while I'm projecting it for you guys right now. Okay, cell cycle. So the cell cycle is very closely linked to replication, transcription, and translation because the replication part is in the cell cycle. So the cell cycle, which is going to be two main phases. One of them is called interphase. The other one is called mitosis. The whole purpose here is that you're going to undergo the cell cycle to either grow, develop, or repair any parts of your body. So the interphase portion is subdivided into three parts. It's G1, S, and G2. And what you guys will notice about interphase, if we're looking at this like a pie chart, all of the part that is in red, that's all interphase. So the majority of the time that we're talking about here is interphase. It's like 90%. So G1 is essentially growth. One, you're developing and growing things like proteins and enzymes that you need to undergo synthesis. This is where you have DNA duplication take place, and you're going to make an extra copy of your DNA. Then that leads into G2, which is the creation for specifically the proteins that you need to undergo the process of mitosis. After which phase, now you're ready to enter into mitosis. Mitosis is the main part of the division that's going to end with cytokinesis here. So the stages of mitosis, there's 5. There's prophase, prometaphase, metaphase, anaphase, telophase. So generally when we talk about these, the acronym we use is PPMAT. So it's PPMAT. Prophase, prometaphase, metaphase, anaphase, and telophase. And then last part here is going to be called cytokinesis. That's the last part. So generally speaking, each phase has a couple of steps to them. Oh man, this one's not going to work either. I'll jump back really quick for the purposes of conversation, and then we'll look at the video in class. Prophase is where you're going to condense chromatin into chromosomes. Prometaphase is when you're going to attach the spindle fibers to the centromere of the chromosomes. Metaphase is where the chromosomes line up along the equator. Anaphase is where the chromosomes are going to divide into daughter chromatid. And then telophase is going to be, it's the beginning of the division, which means you're going to be able to see what we call a cleavage furrow. So that's all the verbiage. In class, we'll watch the video and you guys can start to put it all together. So clinical issues with mitosis, cancer, right? A group of diseases characterized by uncontrolled cell proliferation. What does that even mean? So cancer is also called uncontrolled cellular proliferation or division. So what we're saying is there are cells that are dividing rapidly without control, and they end up developing a tumor or a neoplasm. The cancerous neoplasm is going to be called a malignant tumor or malignancy. It has the ability to undergo metastasis and then the spread of cancerous cells to other parts of the body. So While cancer is not good, if you can keep it in one location, as long as it's easy to identify and you can like see all of the perimeter of it, generally speaking, it's easier to get rid of. The really hard part with cancer treatment is when you had it in one area, let's just say like your liver, and then it moved to the esophagus, and then it moved to the spine, and then it moved to the brain, right? It's in too many different areas that are affecting too many different parts of your body that are really critical to life. That's where it becomes really, really fatal. Hyperplasia are going to be an increased number of cell division. This could produce a benign tumor, which does not have the ability to move. Benign is good in a sense because it's localized, it's in one place. It could also create a malignant tumor, which means that it's spreading due to the detachment of the tumor and it's entering into the blood or the lymph and being carried to a different location of the body. How you can identify them is through imaging and tissue samples, and hopefully go from there. What causes cancer is things like carcinogens. So like smoking will introduce carcinogens into your body. X-rays, which is different ultraviolet lights that your body's not used to, or even viruses. So carcinogenesis, which is the creation of carcinogens, is a multi-step process that takes many years and lots of mutations in order for it to occur, which is why so many like teenagers used to smoke like 30, 40, 50 years ago because they had no idea that it was a carcinogen and it would affect their body a long time down the road because it took a long time, many years, for the mutation of their cells to take place in order for them to develop things like lung cancer. There's different types of cancer. So carcinomas are going to come from epithelial cells. Melanomas are going to come from melanocytes, which are pigments of your skin. Sarcomas are going to come from muscle cells. Leukemias are going to be cancer of blood. And lymphoma is the cancer of the lymph. So growth and the spread of cancer cells can divide rapidly and continually, and they can trigger what we call angiogenesis, which is the growth of new networks of blood vessels that would continue the spread. Cancer cells can leave their site and then travel to other tissues, which again would be called metastasis and be harmful. So last part here with the clinical issues in the aging process. Aging is a normal process that gets accompanied by progressive alteration of your body's homeostatic adaptive responses. So geriatric medicine means it's medicine or practice of elderly people. Generally speaking, medical problems arise more commonly just because the body is starting to change drastically. So physiological signs of aging is like gradual deterioration in function and capacity and their ability to respond to environmental stresses. Extracellular components of the tissue also change with age. So some of the theories with aging is genetically programmed cessation of cell division changes, glucose addition to the proteins change, free radical reactions are increasing, and there's excessive immune responses because your body is so prone to getting sick. Okay, you guys, that ends it for today. If you guys have any questions, please let me know in email or in class. I'll see you guys next time.